Okay. So, we at UM conduct a large variety of studies, all aiming at better understanding psychedelics and their clinical applications. And we want to provide you with glimpses of these different aspects of our research in the upcoming 30 minutes. So first, I will start by explaining or by showing what we know about how people use psychedelics and why, and what do they experience. Pablo will follow by sharing what we know about how they work in the brain. Nadia will focus more on a specific mechanism that might be underlying a therapeutic effect. Johannes will talk more about how we can test this therapeutic effect in clinical trials. And Mauro will tie things together by sharing some clinical applications. So first, how and why do people use psychedelics? To answer such questions, we conduct survey studies, which provide an opportunity to collect a large amount of data in a relatively short period of time. And these can further provide a focus, what to focus on in future studies. So first, how do people use psychedelics? People use psychedelics in regular or perceptual doses that, elicit perceptual changes. And when we look at survey data, people tend to use psilocybin-containing mushrooms or truffles and LSD most often. And doses that are typically used include 3.5 grams of psilocybin-containing mushrooms or 200 micrograms of LSD. People also use psychedelics in subperceptual doses or low or microdoses, and these are typically about 10% of a perceptual dose. And people use them repeatedly, for example, 2 or 3 times a week for a certain time period. And here again, LSD and psilocybin are most often used. In typical doses are about 10 micrograms of LSD and 0.5 gram of psilocybin-containing mushrooms or truffles. So we also wondered, why do people use psychedelics, and in this case, microdose? So it seems that the most common motives that people microdose are to enhance your performance, but also to enhance mood, but also to alleviate some symptom relief or symptom alleviation for psychological, but also physical disorders. So it seems that there is a population who use psychedelics to relieve some symptoms for disorders. So when we further investigate this self-medicating population, it seems that they found microdosing more effective than conventional treatments they tried for both mental and physiological disorders. And especially this was the case for people diagnosed with ADHD. So they rated microdosing as being more effective than their conventional treatments. However, this self-rated effectiveness was based on 3 questions. Did it work? Did your symptoms disappear? And did your quality of life improve? And these 3 questions were asked in retrospect, and no validated questionnaires were used in this survey. So to fill this gap, we conducted another survey last year. And this survey had a prospective design, which means that we followed people before they started microdosing, and we assessed them 2 weeks and 4 weeks later. We recruited adults diagnosed with ADHD who had the intention to microdose on their own initiative, and we used validated questionnaires to assess ADHD symptoms and well-being. We included 226 participants and 65 and 46 at each time point, respectively. So we expected to find, based on the previous study, we expected to find decreases in ADHD symptoms and increases in well-being, and this is what we found for the ADHD symptoms. So ADHD symptoms were decreased 2 weeks after microdosing compared to baseline, and were further decreased at the 4-week time point. And well-being was increased 2 weeks after microdosing and remained at this level at the 4-week time point. So These results were in line with what we expected, and we're in line with the previous survey showing that microdosing was indeed effective for people with ADHD. However, using the design that we use, we cannot exclude a possible placebo effect. The participants that we recruited were mainly recruited to a microdosing website, and also these participants wanted to use microdosing to self-treat their symptoms. And both these factors can inflate a potential placebo effect. So if we want to find out if microdosing can be really effective as a treatment for ADHD, but more generally, if we want to investigate if psychedelics really have a therapeutic potential, we first need to understand what really happens in the brain when people use psychedelic. And now I want to give the word to Pablo.
Hello. So I'm just going to talk to you a little bit about the mechanisms of action that we study here at Maastricht. So really, we've known now over the last 70 years that psychedelics are serotonergic 5-HT2A agonists. And we know this from, let's say, knockout studies or using antagonists such as ketanserin. And really, the outcome of this is mimicking the action of serotonergic neurons stemming from the raphe nucleus, acting at deep cortical layers to induce glutamate release. So what is glutamate? Well, it's an excitatory neurotransmitter. And essentially, by binding to NMDAR and AMPA receptors in cortical layer 5 pyramidal neurons, it induces a state of net hyperexcitability. And this gives rise to the global changes in connectivity that we see under psychedelics. But what's interesting about this is that it often follows the pattern of 5-HT2A expression. So studies by guys like Salada or Haijen, they show that if you stimulate this receptor in the medial prefrontal cortex, you actually get glutamate release. So in order to try and understand whether glutamate is a final common pathway, we try to use studies using macrodoses, or as Eline mentioned, doses that wage perceptual effects. And really, this allows us to get more bang for our buck and also access the rich phenomenology that comes with the subjective experience. So this is just taken from Leo Rosman's paper on the depression trial with psilocybin. And what you see is that the respondents in purple actually have more access to sort of a mystical type experience, encompassing feelings of ego dissolution, unity, loss of self. And what the advent of neuroimaging and psychedelics has done in the last 10 years is that we can really relate that to a particular network called the default mode. Now, everyone probably has heard of this place, but just to recap, it's a task-inactive network that, let's say, relates to processes such as mentalizing, introspection, mind-wandering. And what we know is that the subjective experience and changes in DM and connectivity are closely linked. However, we don't really know where glutamate fits in all of this. And so here at Maastricht, we run studies using a range of psychedelics in healthy volunteers, whether it be psilocybin, and now more recently ayahuasca and 2CB, to try and understand the role of glutamate. So in light of the fact that we're currently running studies, I'm going to present to you some findings from a prior research that we've done with psilocybin, in which 60 healthy volunteers were administered a low to medium dose of psilocybin or placebo. And they were subjected to something called magnetic resonance spectroscopy and fMRI at 7 Tesla. So what is magnetic resonance spectroscopy? So it's basically a technique to get a neurochemical snapshot of a particular brain region in order to see the sort of metabolites in that particular place. And in this study, we focused on the medial prefrontal cortex based on prior findings, and the hippocampus due to its involvement in the narrative self. And of course, fMRI allows us to basically look at changes in brain connectivity. So 7 Tesla is just very high resolution fMRI. And of course, we paired this with, let's say, subjective effect markers. So in the lighter color, this is taken from the study, you see that people under psilocybin really do feel feelings of ego dissolution, as demarcated by OB, so oceanic boundlessness, AED, anxious ego dissolution, and ED, ego dissolution inventory, another measure. The others being visual and somatic effects of psychedelics. So what was really found? So in terms of connectivity, so here, this is now the DMN extracted by something called an independent components analysis. You see that under psilocybin, there is a reduction overall in the anterior and posterior DMN. Whereas for the medial prefrontal, whereas for the glutamate findings, what you see, as I think Jan briefly showed, is that glutamate in the medial prefrontal cortex goes significantly up, whereas in the hippocampus, it gets diminished. Now that we know that we have these two biological markers, how does it kind of tie into the subjective experience? So there's something called a canonical correlation analysis that you can do, which basically lets you predict, let's say, the variance of the subjective experience using these two traits. And what was found is that, so really, the more medial prefrontal glutamate you get, and the less anterior DMN connectivity you have, then really the more anxious or negative is your experience of ego dissolution. Whereas the opposite, for the hippocampus, these diminishments in glutamate and, let's say, accompanying changes in DMN connectivity actually predicted the more kind of blissful feelings of ego dissolution. So what does this really mean as a whole? And for that, you kind of have to look back at prior literature. Is that, so in conditions, so this is a pyramidal neuron, right? But in conditions such as anxiety, what we often see is that there is a dysregulation of this excitatory-inhibitory balance mediated by glutamate and GABA. So it might be that psychedelics in the long run are actually, through receptor downregulation, perhaps restoring this normal EI balance. In regards to the hippocampal findings, well, this is often seen in other disorders, other conditions of ego disturbances. So the hippocampus is part of the medial temporal lobe subsystem. And what we see, like previous findings by Lebedev, this uncoupling of the DMN and the medial temporal lobe actually, give rise to ego dissolution. But we also see altered connectivity in this region in conditions such as depersonalization or schizophrenia. However, as you see, that medial temporal lobe subsystem is only one of many. There are many regions that pertain to feelings of sense of self, etc. So in our next studies using ayahuasca and 2CB, we try and look at MRS in different hubs that are pertaining to sense of self, such as, let's say, the PCC or the anterior insula. So now that we know, sort of, I guess, that glutamate is involved as perhaps in the final experience, how does this actually pertain to the longevity of, let's say, psychedelic effects? How does this relate to the fact that we see these long-term positive outcomes under psychedelics? And I think that's something Nadia can explain.
Hello. So I'm going to continue where Pablo left off. So Pablo stated that psychedelic substances can increase extracellular glutamate, and now I'm going to go one step further because that can also enhance BDNF. And BDNF is associated with neuroplasticity, and that might be one of the therapeutic mechanisms underlying psychedelic substances. So just to start off, what is BDNF exactly? BDNF is brain-derived neurotropic factor, and it's stated it's a protein and is associated with higher levels of this substance in your brain. Your brain will be much happier. It's like a healthy brain, to put it very simplistic, of course. And the BDNF is mostly active in the limbic and prefrontal systems. So just to go back on basic neurotransmissions, so you have here the pyramidal neuron that Pablo already mentioned. So here you have neurotransmitters in the synapse, and it will act on the dendrites, and it goes back to the axons, release of neurotransmitters, and so forth and so forth. So what does BDNF do? BDNF has effect on three processes. Neurogenesis, spinogenesis, synaptogenesis is quite difficult terms, but I'm going to explain all three of them, so don't be scared. So just, again, a very simplistic view of the neuron. The red ones are the dendrites, the blue ones are the axons, and then between the dendrites and the axons, there's a synaptic cleft where neurotransmission takes place. So what is neurogenesis? Neurogenesis means the ability to promote growth of dendrites and new axons. And there's a study done in cell tissue, and they added 3 psychedelic substances and compared it to a vehicle, like a sort of placebo we use in human studies. And they saw that the number of branches on the dendrites and the number of primary dendrites was increased following the psychedelic substance. So that is neurogenesis. Then spinogenesis means that you have on these dendrites, you have these little spines, and they saw that with psychedelic substance. Here, this is the dendrite. You see that these spines are more created following psychedelic substances. And then you have synaptogenesis, the final one. It basically means that there's more synapses created following psychedelic substances. However, just to mention, this is cell tissue, this is brain tissue and cell culture, so this is not found in human studies. But do we know that this also happens in humans? But the thing is, we cannot do this kind of studies in our volunteers. Of course, that would be ethical. And another thing to mention is that these doses of psychedelic substance is actually relatable to the high doses of psychedelic substances used in humans, so the hallucinative doses. But we were interested, as Eline already talked to you about, microdosing of psychedelic substances or low doses, also very increasing in the last few decades, and also with the therapeutic potential people use it for. So we were interested, does a low dose of psychedelic substance also increase this BDNF promotion? So that's what we did. We had three psychedelic doses of LSD, 5, 10, and 20 micrograms. So those are subperceptual. And we compared it to the placebo. And how do we measure BDNF? We cannot cut open brains and measure BDNF there. So we took blood samples. And in blood samples, you can also measure BDNF protein. And we actually found that with the 5 micrograms and the 20 micrograms, as you see here, after psychedelic intake, BDNF was increased compared to the placebo. So this kind of gives an indication that with a low dose of psychedelic substance already can promote this kind of neuroplasticity. However, I have to say that this is preliminary data. Why do I say that? It's because our number of participants is quite low. So but we have a study we just completed with repeated doses of microdosing, and those blood samples still need to be analyzed. So hopefully we can replicate these findings. So what does it mean? Low levels of BDNF is like a not so happy brain, but psychedelic substances might improve this and make your brain happier. Although I have to say this is again in healthy volunteers, so the therapeutic effects of this substance need to be done in clinical trial, actual patient populations, and that is something Johannes will talk to you about.
Thank you very much. Hello. So yeah, we've heard about the reasons for why people use psychedelics by Eline, as described, as for example, as in the self-treatment of ADHD patients to alleviate some of their symptoms. And then we have seen from Pablo and Nadia the underlying biological changes that could cause these improvements, maybe occasioned by glutamate and BDNF changes. But how do we get that into practice? What's the next steps? And these are really the clinical trials. And for that, I'll give you a bit of background. In order to market a medication, it has to go through different phases that Kim already touched briefly upon. There are some steps before and after, and can even be intermediate ones, but these are the three main ones that you have to do in human research in order to bring a medication on the market. And these are really to determine the best dose that you have in phase 1 and phase 2, while that has the largest therapeutic effect, while keeping the side effects and other adverse events at a minimum, so all the risk associated with that. And so in psychedelic research, we have seen already phase 2 and phase 3 trials as well. This phase 2 trial is actually being also, for which we in Maastricht also study center, which Kim is coordinating. But we are especially also interested in understudied substances and novel psychoactive compounds. So that we've done several phase 1 studies in substances such as 4-fluoamphetamine, JWH18, a synthetic cannabinoid, and mitragynine, the main alkaloid of kratom, a plant from Southeast Asia with analgesic properties. And then my research focused on 5-MeO-DMT, which I'll tell you a little bit more about now. So what is 5-MeO-DMT? It is it's a tryptamine closely related to psilocybin and DMT, found in a variety of plants and at least one animal species, the Sonoran desert toad. And 5-MeO-DMT has three very distinct components. One, that it has very rapid onset. Usually it's smoked or so inhaled, so it works basically immediately. It's a very short duration of about 5 to 30 minutes, and it's very potent. And what do I mean with highly potent? It is especially good in eliciting so-called peak experiences or mystical experiences. These have, for example, been shown in psilocybin trials. That at a certain dose, participants reach this state of ego dissolution or feelings of oneness with the universe. And these are experiences that are very profound and meaningful and can even be very transformative. So these might be the experience that could elicit therapeutic changes far beyond the actual workings of the drug. So when the drug has left the system, you can see changes, positive effects long after that. So we wanted to really test this and put it into, to see how 5-MeO can work with that and how we can implement that. So first we did that study in healthy volunteers just to show that the doses that we use are safe to administer, and then proceeded with a trial in patients with treatment-resistant depression. So there we recruited 16 patients and had a 2-part setup. In part A, we had 8 participants who received single doses. So they had 1 dose on the test day, and the main focus of this part was safety. And throughout the entire study, we followed the participants for 1 week only, because the main focus was safety. And then we had a second part with another 8 participants where we focused more on the efficacy. And here we administered up to 3 doses in 1 day of increasing magnitude. So it was a medium-low dose, a medium-high dose, and then a high dose. And we did that in order to find out the best dose to elicit a peak experience. So we had this, the setup of potentially 3 doses, but we only administered the subsequent doses if the previous one did not elicit a peak experience. What were the results that we found here is mainly that, so these are only the top-line results, that throughout the study we saw that it was very safe to administer not only the single doses in part A, but also that in part B, if we administered several doses, there was not a larger increase of adverse events. We didn't see any serious adverse events or any clinically significant changes in vital signs. And then most importantly, or the most interesting, the efficacy aspects, especially of that part B that we're looking at for these 8 participants. We saw that 7 of 8 went into remission by day 7, and we saw an overall decrease of symptom severity by 76%. And these are very, very significant numbers that we are ourselves very astonished by. And we are happy to move forward with these results. So we see that there can be symptom improvements and symptom alleviation in patients in research, but how do we actually implement that in clinical settings? Because that would then be the next step once we get to legalization or implementation of these substances. And that was what Mauro will tell you about.
Okay, so speaking of clinical outcomes, we are obviously interested in learning what fits the patients best and what generates the most clinical improvements in them. And obviously what we know is that the changes in, for example, in clinical effectiveness are sustained by this window of opportunity that psychedelics seems to open. And this window is supported by increased cognitive flexibility, as my colleagues just said, by increased neuroplasticity and by increased suggestibility, which we could define as the degree to which we are open to new ideas, new thought patterns, new behavior, and new self-representation. There is quite an established consensus concerning the importance of set and setting, but what we are really still looking for are theoretically grounded models to conduct psychedelic-assisted therapies. Out there are many proposals about this, but we need to still build a strong foundation for this endeavor. So what we do was embarking into a literature review to find out what is currently being done in clinical research with psychedelic-assisted therapy, and we cast a wide net in terms of search terms in order to get the most of the studies that deal with this important topic. And we also cast a wide net in terms of the substances that are being used. We included both classical psychedelics, such as LSD and psilocybin, but also atypical psychedelics like MDMA and ketamine, given the importance that the literature is giving to these compounds. We ended up with 55 studies, and after a quite intensive selection process, and today I would like to talk to you about what we found in terms of therapeutic models. First thing we found is that there are basically 2 kinds of models: ad hoc models, which are models that were explicitly designed to work with psychedelics, and adapted models. We won't go through all of them, so don't be scared of this, but just to give you a general impression of what are the open issues in the field. We will pass through some of them. So these are some of the ad hoc models. I'm sure you will recognize some of them from the literature you may already have read. And then we have adapted models in which you can see very established models of body-living psychotherapy, such as the CBT and MET and such. And what we learned by scouring this literature was that basically all models go through the same structure that Kim was talking about before. So preparation, drug session, and integration. But within these boxes, there is a huge amount of variability, both in terms of duration, as you can see, but also in terms of the principles that these phases actually use. For example, there is not a clear guideline on what to discuss with patients during preparation. Obviously, if I'm working with a traumatized patient, I will inquire about trauma. And if I'm working with a depressed patient, I will inquire about depression. But what about other classical topics of psychotherapy, such as family relationships, thought patterns, and other aspects? Drug sessions. So obviously in psychedelic psychotherapy, you may not know this, but we use psychedelic drugs to induce therapeutic change. And here, again, the variability in duration is wide, and it depends on what kind of a substance you are intending to use. But there are two great open questions here. Who's driving the experience? I mean, shall I strive to grant my patient an uninterrupted journey where his unconscious mind or her unconscious mind can freely roam the mystical experience, or should I guide the experience based on my clinical hypothesis on my patient? And I will say a little bit more about this in the coming minutes. And then integration. So everybody values integration, which is a great thing. But again, we don't really know what we should do there. There are some general ideas, but we lack a strong theoretical model to do this. The idea is to generalize and consolidate the learning and the insights that happen during the psychedelic experience into everyday life so that the patient can really, really benefit from the experience. So, moving on to the more general open issues, we must understand what is the role of non-specific themes. In simpler terms, should I talk about your family relationships? Should I talk about your past? Should I talk about your belief, about your life, or the things that you would like to change in your experience? And obviously, it is obvious to think that I must talk about your motivation to change, your etiopathogenetic hypothesis, the hypothesis on the origin of your depression, on the origin of your discomfort, or on the origin of your desire to develop yourself inside a psychedelic-assisted psychotherapy. What about the existential issues? How much time should I give as a therapist to the meaning-making, to this seemingly unique aspect of psychedelic-assisted psychotherapy that really makes you dive directly into the meaning of life and the meaning of your existence, and so on. We could go on mentioning more other themes. And then, in the preparation phase, when we are describing why the treatment works and when we are describing how your, for example, psychopathology work, am I actually describing it? Am I doing psychoeducation? Or am I using suggestion here? Again, in simpler terms, when I'm talking about inner healing intelligence, what am I actually doing? Obviously, I'm assuming, for example, its importance, its relevance, and its value, so that I discuss it in therapy. But we don't really, given the heightened suggestibility that some substances seem to produce, we must understand what we are actually touching in the patient when we are talking about these themes. And then there is the directiveness dilemma. Again, who's driving and who's making meaning? How much time should I give to interpretation, to therapist interpretation inside the psychedelic-assisted therapy context? Basically, these are the questions that we must answer if we want psychedelic-assisted therapy to become a really modern, established, and evidence-based approach. And I would have never thought that I would talk about psychedelic drugs inside a church. And as an Italian guy, I'm quite worried that I'm going to be shocked to death once I go out. But since we are talking about mystical experiences, I hope you'll forgive us. And now I'll leave the words to Johannes to wrap this up. Thank you.
Yeah, thank you very much. Thank you. Just to give you a quick outlook on the future directions that we have, so we want to, of course, continue with our clinical ADHD microdosing trials. longitudinal LSD microdosing trials just to really elucidate the effects of, for example, BDNF. We're going to elucidate the mechanism of novel psychoactive substances to broaden our horizons on also what Jan mentioned, these maps that we want to include the metabolomics, for example, and also to see the difference between novel psychoactive substances and the classic ones. And move forward with the 5-MeO-DMT studies or other clinical studies in patient populations. And also want to look at the treatment of chronic pain using psilocybin, especially with the background of how does suggestibility influence these effects. And so this is the group that we have in Maastricht, the Ph.D. students, and we're very happy to show the research that we have been doing and that we are doing and that we plan to do to you. Thank you.
Thank you very much to everyone. Another round of applause now that everyone is back on stage. Perfect. I do believe we still have time for a few questions. Also, as a reminder for the people online, now is your chance to submit questions.
All right. Yeah, thank you, all of you. Very interesting talks. Oh.
Okay, is it working? So we have a question from one of our online participants, more like a question and a remark. So it's regarding that part of the presentation with the fMRI studies. She's wondering about the amount of psilocybin, so only 0.17 milligram psilocybin. That's less than what was used as some active controls. I'm surprised that that is enough to induce effects like oceanic boundlessness. Yeah.
Well, it's dose dependent, right? So in this case, literally all the markers we had were, well, in the end, significant. And participants, at least in the scanner, they were very much eliciting. And when you think about it, so it's body, it's still not, yeah. It's body weight adjusted, right? So in the end, we do have this like consistent effect that we came across for everyone. But we're repeating this actually. So in our new study, we're actually going up a little bit higher. So now here we're going with a fixed dose of 15 milligrams of psilocybin. But we don't expect there to be differences in terms of whether people actually get ego feelings, degrees of ego dissolution or not. Really. Yeah.
Thank you.